All right, thank you, Rasis, for the introduction, and thank you, everyone, for showing up early. I know it's not that easy to get up early these days with the daylight saving time just starting. So thanks for being here. So today I'm going to talk about the two unknowns of nucleosomes, formation and removal. I know the title sounds kind of ambitious, but don't expect too much biology out of this talk. So first, as you can see from the title of this talk, nucleosome is going to be at the center of this talk. So first, I'm going to give you some introduction to nucleosomes. So nucleosome means particles in the nucleus. And physicists would like to name it nucleon, because physicists like to use ON to name particles, whereas biologists like to use OME to name particles. Right? So it just means particle in the nucleus. So when I think about nucleus, what comes to my mind is this very delicious dish. So many of you recognize what this is. It's basically bacon wrapping scallop inside. And this is very similar to nucleosomes in many aspects, right? So as you all know, nucleosomes are basically beads on a string type of structure you can find in eukaryotic genome. So you start from this naked DNA, which goes through nucleosome formation and then higher order structuring inside the nucleus of a cell until you get this X-shaped chromosomal DNA. Well, the biggest difference between the two is that this, of course, is protein wrapped in fat, whereas this is protein wrapped in nucleic acid. Right? Some people tend to think this is protein, what's wrapping the scallop, but I think it's just fat and salt. Okay, so outline of the talk. I'm going to divide my talk into two parts. The first part is going to be about DNA looping, which has to do nucleosome formation. And the second part has to do with how gene expression can be achieved from packaged DNA. And therefore, we're going to introduce a molecular mechanism of nucleosome removal. Okay, so the first part, DNA looping. So before we move on, I would like to introduce the physical concept related to looping, and that is called the persistence length, which is the length scale that is used to characterize the bending stiffness of a polymer. So imagine if you have a flexible polymer like this, you can put 2 points on the contour of this polymer, and then you can also draw vectors that are tangent to the contour of the polymer at those 2 points, and there is going to be an angle between the two vectors. And the size of this angle is going to depend on how far away these 2 points are along the contour of the polymer. So if you take cosine of this angle, it's going to decay exponentially with some characteristic length scale called the persistence length. Okay? So L is a contour length between the 2 points, and LP is a persistence length, which is the property of the polymer. Just to give you some examples, a single-stranded DNA is known to be very flexible. So that means it has a very short persistence length, which is about 2 nanometers. And that corresponds to 7 nucleotides. Double-stranded DNA, because it forms base pairing between two single-stranded DNAs, it's stiffer than a single-stranded DNA. Therefore, its persistence length is longer. And it's about 50 nanometers, which corresponds to 150 nucleotides. Okay? So shorter persistence length means flexible, larger persistence length means stiff. Some macroscopic examples, since this is a breakfast club seminar. So this is spaghetti noodles, and if they're uncooked, then they are very stiff, as you know. Persistence length is about 10 to the 16 meters. So that means you can imagine a very long spaghetti noodle, which is on the order of 10 to the 16. It's going to stay pretty straight, even on that long length scale. Okay, and you can think about ramen noodles, and they're very curly. And if you measure, how much they curve, and they curve on the order of a centimeter or so. So you can think of this persistence length of ramen noodles at 1 centimeter. Okay, but the persistence length that is explained here is a little different than the concept that I'm going to focus during this talk, because this refers to the static persistence length. It's the intrinsic curvature you can find in this structure, whereas the persistence length here is the thermal property of the molecule. Right? Some numbers are going to be important throughout this talk, so I'm going to talk about the dimension of the nucleosome and bending energy stored in a nucleosome. So it's known that about 140 base pair of double-stranded DNA wraps around this protein about 2 full turns, so 1.7 turns. So this is going to be the circumference, right? The contour of the DNA that surrounds the protein molecule. So if you divide it by 2 pi, you can recover the radius of this particle. That's about 4.1 nanometers. You can also estimate the bending energy stored in this DNA based on this persistence length and the radius of curvature. So you apply this elasticity theory, and then you achieve about 70 kBT, where kBT is Boltzmann constant and T is the room temperature, right? So as you can see from this number, this is a very energetically costly process. You need about 70 times the thermal energy to induce bending in this nucleosome. Okay? So that's why we call this very sharp DNA bending. Okay? It's not likely to occur just under thermal excitation. So that also implies that the interaction between the DNA and the histone surface is going to be very strong. So I forgot to mention that there are 4 different colors in this protein complex, and that's because there are 4 different kinds of histones that make up this protein complex inside the nucleosome. And they are named H2A, H2B, H3, and H4. And there are 2 of each in this nucleosome particle. So what's interesting about nucleosome is that they position neither randomly nor regularly. So if you take a genomic DNA and map the positions of the nucleosome, you find that there are some regions that don't like to form nucleosome, whereas some regions like to form nucleosome. As a physicist, then, we tend to think this is because of the mechanical properties of the DNA are not uniform as a function of sequence. So for example, some DNA sequences are going to be stiff, and some DNA sequences are going to be flexible. And that's why you see this non-uniform nucleosome formation as a function of DNA sequence. So we like to really investigate whether this is true. So I'm showing here you a plot that combines 2 different data sets. Well, actually, it's 1 data set that is compared with theoretical prediction. So what's shown here is nucleosome affinity as versus persistence length. So Widom and Siegel a few years ago did a very important study using synthetic DNA molecules. So they assembled about 10,000 different DNA sequences and mixed those DNA sequences with histone proteins and measured the affinity of all these molecules. So what's shown here on the y-axis is the affinity measured from that experiment. And this is log 2 based, so from -4 to 4 here, it's about 400-fold different. And then on the x-axis is the persistence length of that DNA sequence molecule predicted from an independent study that measured persistence lengths of all possible dinucleotides. Right? So as you know, there are 4 different alphabets in DNA sequence, A, G, C, T. It turns out that you can form 10 unique dinucleotide sequences. So you can basically take more than 10 DNA sequences and measure their overall persistence length, which is going to be some sort of combination of the persistence length of dinucleotides that compose the entire molecule. Based on that, you can extract the persistence length of those dinucleotides. I'm not going to get into detail of how you do that calculation, but simply speaking, if you know the sequence of the DNA, you can count all the dinucleotides and just retrieve the entire persistence length. So that's what's plotted on the x-axis. And as you can see here, you don't see a very strong anti-correlation, which you would expect if nucleosome formation really depends on the mechanical properties of the DNA. Right? So anti-correlation means that if you have a very soft DNA molecule, you're likely to form a nucleosome very well. Right? So you would see points distributed along that axis. However, you don't see that. Points are just scattered all over the place. I'll give you one extreme example. Right? So you can pick the DNA sequence that has the highest nucleosome affinity. Right? So that's why we name it nucleosome DNA. and you can also pick another point in the exact opposite. So we call it antinucleosome DNA because it has the lowest nucleosome affinity. Right? So between these two sequences, you actually see that the model predicts that this DNA sequence, which doesn't form nucleosome very well, it's actually softer than the DNA sequence which forms nucleosome very well. Right? So here you'll see that the model prediction and the experimental data, they don't agree with each other. Right? So, and it's known that this study also admits that this model prediction doesn't work well for repeat sequences or sequences that have intrinsic curvature. So as a physicist, we like to think of nucleosome affinity as having something to do with the intrinsic stiffness of the DNA. Right? So if you see a 400-fold difference in nucleosome affinity, you can apply the Boltzmann factor to extract the bending energy difference between these two DNA molecules, and it comes to be about 6 kBt, right? 6 times the thermal energy. So the question is there an independent method to measure this number, the difference in bending energy between the two DNA molecules? So the methodology we use is called single molecule fluorescence resonance energy transfer. Some of you might be familiar with this technique. Let me just explain it very briefly. If you have a molecule that's on the order of 5 nanometers, well, to be more accurate, if there's a molecule that moves on the order of 5 nanometers or so, you can put 2 different dye molecules in the same molecule, right? And they are called donor and acceptor. And the reason one is called donor and the other is called acceptor is because the donor can transfer some of its energy to the acceptor, and donor is the molecule that is directly excited by the laser or your excitation, and then it's going to fluoresce by itself when the 2 dye molecules are far apart from each other, further than 5 nanometers typically. However, when the distance between the two dye molecules becomes smaller than 5 nanometers, then, like I said, there's energy transfer from the donor and acceptor. Therefore, the acceptor starts emitting fluorescence. Okay? So by looking at the relative fluorescence intensities, you can extract what's happening to the distance between the 2 dye molecules. So this, as you can see, is a very powerful method to monitor conformational changes of biomolecules at nanometer length scale. So this is our experimental design, which is very simple. So we're going to take about 190 base pair long DNA molecules, which is about 60 nanometers. And you can compare that to the persistence length, which is about 50 nanometers. Right? So this DNA molecule is pretty short and is pretty stiff. And once in a while, this DNA molecule is going to form this loop. So a perfect technique to detect this is obviously fluorescence resonance energy transfer. So we put 2 dye molecules on both ends of this DNA molecule. And then when the DNA is not looped, meaning the distance between the 2 dye molecules is large, then we only see fluorescence coming from the donor molecule. However, when the DNA molecule loops, then the distance between the 2 is very short, so we start seeing fluorescence from the acceptor molecule. Okay? Now, the trick here is that even when the DNA molecule forms a loop, it's going to be very short-lived, right? So these 2 ends are going to diffuse away in nanoseconds, so we cannot really detect whether a loop formed or not. So we introduce complementary sequences here so that when the 2 ends come close to each other, there's a high probability that this base pairing is going to form. Okay? So that's how we stabilize this loop state to detect it. And this is just to show you that we have this experimental protocol to construct any arbitrary DNA sequences that have all the necessary elements for surface immobilization and FRET detection. Right? So we start out with this DNA sequence that we are interested in, and we perform 2 separate PCR reactions, and then we melt the DNA and do a strand exchange, and then cool it, and this is your final product, and it has the biotin, which is what used for surface immobilization, and the acceptor dye molecule at one end and donor molecule at the other end, with this complementary base pairing region. And the setup is very simple. We use a standard total internal reflection microscopy to detect FRET signal coming from single molecules immobilized on the surface. And this is the signal that we're going to expect to see as a function of time, depending on whether the DNA is looped or not. So if the DNA is looped, we expect high FRET signal. When the DNA is unlooped, we expect low FRET signal. Right? So this FRET fluctuation tells you the looping kinetics of this DNA molecule in real time. So as I mentioned before, we're going to compare two different DNA molecules which have opposite nucleosome affinities. So one is called antinucleosome DNA, the other is called nucleosome DNA. As you can see, these are repeat sequences. So these are synthetic DNA molecules that whose sequences are obtained from this landmark paper from Widom and Siegel in 2009. Now, what's different between these two sequences is in the antinucleosome DNA, you see these AT-rich regions, which are about 5 base pair long. Right? So they are totally composed of A's and T's. As you know, they have very different properties than G's and C's. So overall, we can say that the AT content in the antinucleosome DNA is much higher than that in the nucleosome DNA. So this is our typical images. So as you can see, when donor intensity is high, acceptor intensity is low, and when donor intensity is low, acceptor intensity is high. Right? So this anti-correlation means that there is FRET fluctuation as a result of distance change between the two ends of the DNA molecule. Okay? So from these FRET signal fluctuations, we can extract dwell times spent in these different states. Right? So there is loop state and there is unlooped state. So you basically take all these times of the events that are observed in either the looped or the unlooped state, and you can do some sort of analysis which is very standard in single molecule biophysics called dwell time distribution. So you just take all these dwell times and plot a histogram. Okay, so how many events are in a given window. And normally in a very, for example, in a two-state system, you would see a single exponential decay from one state to the other. However, when you try to fit this to a single exponential, it doesn't fit very well. It doesn't fit even to a double exponential. We see that it fits well to a stretch exponential. And we have models to explain why that is, but I'm not going to get into the details because it's a topic of another talk. So what's important is that we can extract the mean dwell time spent in each state, and those are called lifetimes. So what's plotted here is the lifetime of the DNA molecule in the unlooped state, and this plot is the lifetime of the DNA molecule in the looped state. As you can see, the lifetimes of antinucleosome DNA and nucleosome DNA are different in those two states, so this is a good sign because now we can use this kinetics measurement to say something about the differences between these two DNA molecules. So I'm going to come back to these figures later on during the talk, but for now, let's just focus on what sequence feature gives rise to this difference. So as I mentioned, antinucleosome DNA has these AT stretches repeating every 10 base pairs, and it's known from this large-scale study that among all fibers that you can assemble, it is these fibers that are very rich in A's and T's that don't like to form nucleosomes very well. So we thought that maybe these AT stretches are very important. So what we did is we switched the middle base of these AT stretches from T to G. Okay, so we don't have those AT stretch anymore. And we did the same kinetics measurement. So first, focus on these two original sequences, antinucleosome and nucleosome, antinucleosome and nucleosome. Okay? So those were the sequences that we tested a few slides ago. And then we are adding this new modified sequence where we change the T in the AT stretch to a G, right? So this is labeled as antinucleosome T to a G. As you can see from this plot, now this modified sequence behaves more similarly to the nucleosome sequence than the antinucleosome sequence, right? So the key point here is that although overall sequence is similar to the antinucleosome DNA, in terms of behavior, it is now much similar to nucleosome DNA. Okay? So that means that whatever this AT stretch does, it's very important in determining the kinetics of DNA looping. So now let me go back to these lifetime plots, and we can compare the antinucleosome DNA and nucleosome DNA. And I'm going to conclude something about the stiffness of these two molecules. Okay? So how do we relate stiffness to the time it takes for a DNA molecule to loop? Right? It's very simple. In the elastic regime, if you have a very stiff rod, for example, it's going to be very difficult to form a loop. Okay? So that corresponds to a very long lifetime in the unlooped state, meaning it takes longer to form a loop. And that's what we can use to conclude something about the stiffness difference between these two molecules, right? So if you compare the lifetime spent in the open or the unlooped state, then nucleosome DNA spends more time than the antinucleosome DNA, right? So what can we say about the flexibility of these two molecules? Well, we can say that the antinucleosome DNA is more flexible, okay? And this is a complicated way of saying that. If you have a stiff molecule, you basically have a very, very curved harmonic potential, right? So you have this steep slope, which raises the energy barrier that you need to overcome to get into the loop state. And remember, the loop state is stabilized by the base pairing interaction, so that gives you another harmonic potential energy well here, right? So the difference between a stiff molecule and a flexible molecule is basically the height of this energy barrier. In a flexible molecule, the energy barrier is lower, therefore it takes less time for you to cross over the barrier. So from that first plot, we conclude that antinucleosome DNA is more flexible, okay? And it makes sense. However, when I give you the second plot, which is the lifetime of the DNA molecule in the loop state, okay? First of all, some of you might question, okay, why is the lifetime of the loop state any different between the two DNA molecules? Because the loop state is stabilized by this base pairing region, which is commonly shared between these two DNA molecules. Okay? So the answer is because when the DNA molecule is looped, there is a restoring force stored in the DNA molecule due to bending. Right? So this is like, closing a very stiff spring into a loop, and the loop is going to try to open up because there's this restoring force. So if you think about this restoring force, then you can figure out that a stiffer DNA molecule will tend to open up faster, right? Because the restoring force is larger. So based on this plot, antinucleosome DNA spends less time in the loop state, which means that it's actually the stiffer of the two, right? Antinucleosome DNA loops faster. And again, you can introduce this energy landscape argument, a complicated way of explaining this. So you have this energy well, which represents the base pairing interaction. And for a stiff DNA molecule, then the slope is very stiff here, which basically corresponds to a lower energy barrier to cross over, okay? So which leads to faster looping. So based on that second plot, then we have this result, which is antinucleosome DNA is stiffer. Now, if you have an attention span that is longer than 3 slides, then you remember that I just said that antinucleosome DNA is more flexible 3 slides ago. And now I just said in the previous slide that it's stiffer than the nucleosome DNA. So what's happening? Antinucleosome DNA can't be more flexible and stiff at the same time than the nucleosome DNA, right? So we hypothesize that maybe the initial starting point in looping transition is not the same between these two DNA molecules. Okay? So what that means is we assume that the DNA in the unlooped state is straight. It always starts from the straight state, but maybe one of the two molecules is intrinsically curved, so it starts from a different initial point, right? So you start from a point where the distance between the two ends is closer, and maybe that affects the looping time we measure. Okay? So the assumption or the hypothesis is that the stiffer DNA molecule is also more curved. Okay? So even though it's stiffer because the distance between the two ends is closer, you can still loop faster, contrary to what I just explained a few slides ago. So we set out to test this by using gel electrophoresis. The idea is that if you run gel electrophoresis at very low temperature, then you can stabilize a DNA molecule in the open state, right? So we are not really looking at the effect of bending here. We are just looking at the intrinsic curvature or permanent curvature of the DNA molecule. So although all these three DNA molecules have exactly the same length, we see a very different gel mobility. And this is very well known that gel mobility represents the intrinsic curvature of the DNA molecule. So the molecule that runs slower on the polyacrylamide gel is more curved than the molecule that runs faster. Okay? So this is the curvatures we can derive from this gel mobility shift assay. Okay? So conclusion is basically antinucleosome DNA is more curved. And although it's stiffer at the same time, it can loop faster. And we do some basic calculation, right? So starting from this Fokker-Planck equation. Okay, before I go on to the details, so what did we learn so far? We learned two things. We have two DNA molecules, and we measure the looping times, right? The time it takes for the molecule to loop. We also measured curvatures of these two molecules. One is more curved than the other. So based on these two measurements, can we then extract the expected bending stiffness of these two DNA molecules, right? So from looping time and curvature, can we extract the bending stiffness? And there's a way to do this, and it's called this mean first passage time calculation. And it starts from this Fokker-Planck equation, which is also known as Smoluchowski equation. And it's basically describing diffusion in the presence of a potential. Okay? So here, the time evolution of probability density is going to be related to the variation in the probability flux, J. Okay? So this equation is called the constitutive equation. And then you can relate this probability flux, of course, to diffusion term and the potential term, right? So in the absence of potential, this is just random walk. And then you have this potential because the DNA molecule has bending energy as a function of end-to-end distance between the. All right, so using these two equations, then you can extract this mean first passage time as a function of all these parameters. Although this is a three-dimensional diffusion, if you use the end-to-end distance as your variable, you could transform it into a one-dimensional problem. And this is, of course, classical work published many years ago by Klaus Schulten. What we don't know, however, is this potential energy, Ux. Okay? So potential energy of this DNA molecule as a function of the end-to-end distance. And the reason it's very complicated is because although I keep describing DNA looping as this, right? I mean, this is not all that's happening, right? You can get looping like this. And so they're all possible conformations. Okay, so even with the same end-to-end distance, you can imagine that there are going to be many states, which means that there's entropic effect coming into play. However, because the DNA molecule we are looking at is very short, well, it's not very short, but it's on the same order as the persistence length, so we can assume that it's pretty stiff, meaning that it's not going to go through many curves and wiggles. Okay? So we assume that the molecule is going to be the curvature is going to be uniformly distributed, and then this is a trajectory that's going to take most of the time. Right? And if that's true, then the potential energy could be expressed as a function of this angle theta, right? So it's basically a harmonic potential. And then because we need to introduce intrinsic curvature, right? The starting point is different, so that's this theta naught, which is subtracted from the theta. And then theta is related to the distance between the 2 points, right, which is x, by some basic algebra. So we can then approximate the potential energy as a function of end-to-end distance in this simple form. And we plug that in and do some number crunching, and I can tell you that as a result of this, we find that antinucleosome DNA is 1.4-fold stiffer than the nucleosome DNA, which makes sense based on how we name these DNA molecules, right? Antinucleosome DNA is a DNA molecule that doesn't like to form nucleosome. Therefore, it makes total sense that it is the stiffer of the two by a factor of 1.4-fold. Okay? And we already know that based on the average persistence length of the DNA molecule, the energy stored, the elastic energy stored in the nucleosome is about 70 times the thermal energy. And then using these two numbers, we can estimate the bending energy difference between the antinucleosome DNA and the nucleosome DNA, which is about 7.4 times the thermal energy. And then you apply the Boltzmann factor, and you get at this number, which is is still larger than what's measured, right? So the number 400 is the nucleosome affinity difference between the two molecules, but now we get this number 1700, which is about 4 times larger. Okay, but again, this approximation is not very accurate, so we expect that this difference is not very surprising from the viewpoint of an experimentalist. But the key point here is that although nucleosome formation is a pretty complicated interaction, right? It has bending energy, electrostatic energy, and in some cases there's might be hydrogen bonds forming between DNA and the histone octamer. We can still narrow this down to the elasticity problem, right? So even if you only consider the elastic energy stored in the DNA molecule, you can reach the similar order of magnitude. And so this is a summary of the first part. We measure DNA looping kinetics by a single molecule fret, and the length of the DNA molecule is about 190 base pair long, 90 base pair. And that length scale looping time is sensitive to curvature. So you cannot just ignore curvature effect. You need to take both into account when you try to extract the looping time. And I also showed you that this A stretch seems to be doing something funny because it's very stiff and curved than other normal sequences. And we would like to understand more about this. And then I also did some approximation to show you that the nucleosome affinity can be in part explained by the estimated bending stiffness. The second part of my talk is moving one level up. So I'm going to talk about how gene expression occurs from packaged DNA and how this is related to nucleosome formation. Well, how this is related to nucleosome removal. So what is gene expression? Now all of you know central dogma molecular biology, which tells you that the information flows from DNA through messenger RNA to protein. So if you have gene on the DNA, then through these two processes, you can synthesize proteins. So these two processes could be lumped up as one process called gene expression. So that's the term that I'm going to use throughout the rest of the talk. You might question, how is gene expression initiated? It's initiated by these transcription factors, which are other proteins that are transported into the nucleus under stress. Right? So genes are not always turned on. Some genes are off, and they are only turned on in response to some extracellular stress. So for example, if you starve yeast cells for phosphate, then there are transcription factors called FOL4, which enter the nucleus and bind the DNA to activate gene expression. So this is how we physicists like to approximate gene expression. Transcription factors coming in and proteins going out. So you can define this input-output relationship for gene expression, where input is the transcription factors and the output is the protein synthesized from the downstream gene. And you all know that there is a promoter region upstream of a gene where transcription factors interact. Okay, so this relationship, known as gene expression pattern or gene regulation function, is going to be a specific function of this promoter, which means that if you swap this promoter with some other promoter, then you're going to see another relationship. Okay? So you might ask, what determines the gene regulation function? Well, it's going to boil down to what happens at this promoter level between the transcription factors and the DNA. How transcription factors interact with the DNA is going to determine the shape of this gene regulation function. So, what determines the interaction between the two? Well, first, the amount of transcription factors, right? The concentration of transcription factors are going to determine the occupancy of transcription factors, and also location of transcription factor binding sites. The ones that are located closer to the gene will have a stronger effect on gene expression. And the affinity of binding sites, right? Not only the concentration of transcription factors, but also the affinity. How tightly the transcription factors interact with those sites is going to determine the gene regulation function. So as you know, the amount of transcription factors is called trans factors because it doesn't reside on the same DNA as a gene, whereas these two factors called cis elements because they are on the same DNA as a gene itself. But now, knowing that DNA doesn't exist naked in the nucleus of the cell, we have to consider the effect of nucleosomes, right? Because in the end, nucleosomes are going to compete with transcription factors. Therefore, knowing the exact positions of nucleosomes is going to be very important in understanding this quantitative gene regulation function, right? So accessibility of binding sites or positions of nucleosomes, which are often ignored, have to be taken into account. And these three factors, we call it the promoter architecture. Okay. So our interest is in understanding how promoter architecture contributes to quantitative gene expression pattern or the gene regulation function. And since we are interested in how nucleosomes affect gene regulation function, we need to know something about what happens during transcriptional activation. So just as particle physicists have their standard model of particle physics, we have the standard model of transcriptional activation. So this is a model promoter called Fo5. The name is not very important. What's important is that there are three well-positioned nucleosomes in the promoter region. Okay? And what's shown in red and green are the transcription factor binding sites. So sometimes they can have different affinities because they have different sequences. And then some transcription factor binding sites can be located outside nucleosomes, like this one, and some are located inside of nucleosomes, like this one. And also there is the Tada box, where transcription machinery assemble for transcription initiation. And it's often covered by a nucleosome for a lot of stress-induced genes. Okay? So what happens under stress is that transcription factor, in this case called Fo4, is going to be transported into the nucleus, and it's going to recruit chromatin remodeling complex to the promoter. Okay? So the first transcription factor binding site it sees is in the nucleosome-free region. So it's going to bind there, and the nucleosome complex is going to bind to the neighboring nucleosome. What happens after that is not clear, but the end point is that the Tada box is going to be exposed. Okay? So nucleosomes are cleared from the promoter so that general transcription machinery, including the RNA polymerase, can assemble and perform transcription. Okay, so how do we measure the gene regulation function? So we have to somehow be able to measure quantitatively the amount of transcription factors and the amount of proteins expressed. So we use fluorescent proteins to do this. So the transcription factor, which is Fo4, is labeled with yellow fluorescent protein, and the downstream gene, which is activated by Fo4, is replaced by another fluorescent protein, CFP. And the transcription factor input level can be controlled by this drug called doxycycline, In other words, this is placed under a drug-inducible promoter. So what we can achieve from the system is that we have a two-color strain, yellow fluorescent protein representing transcription factor input, cyan fluorescent protein representing the output protein. And we can continuously dial up the concentration of transcription factors. What you expect at low transcription factor concentration, not much gene expression. But as you increase the transcription factor concentration, you're going to accumulate more and more CFP signal. I forgot to mention that our transcription factor is also special. It has mutations that make it reside in the nucleus all the time, right? Independent of the drug concentration. So that's why this is a powerful system. These are typical images that we get from our experiment. We have another fluorescent protein that tells us where the nucleus is inside the cell. And using that nuclear marker, we can quantify fluorescence intensities coming from the nucleus. So this is YFP, which represents the transcription factor signal inside the nucleus, and this is CFP, which of course permeates throughout the cytoplasm. So we measure single cell fluorescence intensities, and we can put them on this scatter plot, gene expression level versus transcription factor input level. Okay? So differently colored points represent points coming from cells grown under different doxycycline concentrations, right? So at very low doxycycline concentration, all points are here, and as you increase the doxycycline concentration, you start to accumulate points with higher transcription factor level and gene expression level. Okay? So this curve looks very noisy, and most of the noise is real, meaning it's biological, and has both extrinsic and intrinsic noise contributions. But the mean values are still meaningful. So you can try to fit this to a function which is very often used by biologists called the Hill equation. And this is useful because it gives you two important parameters. So one is called threshold, which basically tells you the amount of transcription factors you need to activate gene expression 50%. And then it also gives you maximum expression level. Right? So this could tell you about the intrinsic capacity of promoter, how much gene expression level it can achieve. Okay, so we are interested in the role of nucleosome in gene expression. And I'll give you a very specific example here. So we generated this model promoter, which has a nucleosome-free region between these four nucleosomes. So there are two nucleosomes on the left, two nucleosomes on the right. Of course, there are more nucleosomes here, but I'm showing you only the four nucleosomes in the promoter region. So what happens upon transcriptional activation is transcription factor is going to bind here. And what's known from previous studies by other people is that nucleosomes get removed, starting from the nearest neighboring region. Right? So these two nucleosomes are removed, and then these two are removed, and then you have an exposed Tata box region. The question that we got interested in is the role of this particular nucleosome here. And the reason is because this nucleosome has an obvious role. It's covering the Tata box region, so it's downregulating gene expression under no stress situation, right? However, this nucleosome here is not harboring anything inside. It's not blocking the transcription factor binding site nor the Tata box region. So we were questioning the role of this nucleosome. Okay? So what is this really doing in terms of gene expression? And generally, people in the field think that more nucleosomes correspond to lower gene expression level, and this makes sense, right? If DNA is highly packaged, then it's going to give hard time to transcription factors accessing it. Therefore, you expect to get a lower gene expression level. Okay? So we like to investigate this using this model system. So how do you study a function of something in biology? Well, you need to delete it and see what the effect is, right? So that's what we are going to do. There are different ways of removing a nucleosome, though, right? So if you have this model promoter, one way to remove this nucleosome is to basically cut out the whole DNA underlying the nucleosome. So you can do yeast genetics, to basically remove the entire DNA which spans this nucleosome. So as a result, you're going to get this promoter, which doesn't have a nucleosome intervening between the transcription factor binding site and the Tata box region. Right? So this we call excision. The other way of removing a nucleosome is obviously you can change the DNA sequence underneath this nucleosome. And if you remember the first part of my talk, I told you that if you have a lot of A's and T's in the DNA sequence, it makes the DNA stiff, therefore it doesn't form nucleosomes very well. So that's what we're going to do. We're going to increase the AT percentage of this sequence, which will destabilize nucleosome formation. So effectively, you can get rid of the nucleosome without getting rid of the DNA. Okay? So these are two different ways of removing the nucleosome. And of course, it comes from this fact that I just told you. If you increase the GC content, then you increase nucleosome occupancy. And this is also shown by this genome-wide computational study by Hughes group at Toronto. Okay, so the first method is excision. So if you excise this DNA, therefore removing the nucleosome, what happens? So what's shown here is gene expression output as a transcription factor input, which was defined as gene regulation function. So what you see is if you remove this intervening nucleosome, then you see that gene expression level goes up. Okay? And that sort of makes sense because nucleosome, in the end, is something that has to be removed from the promoter for transcriptional initiation. So you can think of it as an additional barrier for chromatin remodeling complex to remove. So, giving more challenges to the chromatin remodeling complex will slow it down, make it more inefficient. Right? So that's why you see this gene expression level increase, and we think that it makes sense. However, what's puzzling was that when you remove the nucleosome using the other way, which is to destabilize this nucleosome by increasing the AT percentage of the sequence, we saw that the gene expression level actually dropped. Okay, and so this was a surprise to us at the beginning, but I should be honest that I actually expected this to happen, and that's why we designed this experiment in the first place. Okay. And the reason this drops could be understood if you know what's going on in terms of nucleosome removal. Okay? So this group, collaboration between Clady and Bartholomew, did some very nice in vitro studies. So what's known about chromatin remodeling complex? It's known to be able to remodel nucleosomes, but remodeling doesn't mean removing. Remodeling means that you basically destabilize the histone octamer from the surrounding DNA so you can now translocate the nucleosome, right? So translocation activity of chromatin remodeling complex is known. However, how does that lead to nucleosome removal is not known. So these people did this experiment where they assembled a mononucleosome in vitro. So you take a piece of DNA, you mix it with histone octamers at certain concentration, and you can form single nucleosome on this DNA. And then they threw in chromatin remodeling complex, which was purified with ATP, because chromatin remodeling complex is a motor protein that burns ATP to achieve mechanical work. The end result of this is you get two different forms of nucleosome DNA. So sometimes you get nucleosome, you find nucleosome on one end, sometimes you find it on the other end. However, what you do not see is the removal of nucleosome. Okay? You always see that there's still nucleosome present on this DNA. Okay? So they were puzzled as to why chromatin remodeling complex can remove nucleosomes in vivo when they cannot achieve that in vitro. So they made the experiment more complicated. So now they assembled two nucleosomes on the same DNA, and they repeated the experiments. So they add chromatin remodeling complex with ATP, and they saw the same end result. They saw nucleosome on one end or the other, but the net difference is different here, right? Because you start with a different nucleosome template. You have two nucleosomes to begin with. You have one nucleosome at the end. That means this reaction, we don't know what happened, but it led to nucleosome removal by chromatin remodeling complexes. Okay? So the model is you need more than one nucleosome to get removal. of either of them. And this is the model that is explained in more detail, right? So if you have a promoter that has 3 well-positioned nucleosomes, and this is a transcription factor binding site, and this is a Tata box region, and this is the intervening nucleosome, right, which we were questioning the role of. So what happens is that in transcriptional activation, transcription factor binds here with the chromatin remodeling complex, and this chromatin remodeling complex can bind to the neighboring nucleosome. Okay? And cryo-EM structure of the chromatin remodeling complex is known, and the exact shape can be simplified like this. So it really has this cavity inside which totally fits over a nucleosome. Okay? So what happens next is that there is translocase activity of this chromatin remodeling complex, which leads to peeling of DNA from the histone octamer inside this chromatin remodeling complex, right? So this extra length of DNA has to come from somewhere, and it will come from this neighboring DNA, right? So this DNA gets pulled in to the chromatin remodeling complex. Therefore, this nucleosome, which is over the Tata box region, approaches this complex between the chromatin remodeling complex and the nucleosome. And that has to result in some sort of disruption, right? So what gets disrupted, we think, is this nucleosome that is covering the Tata box region, not the nucleosome which is bound by the chromatin remodeling complex. Okay? So the net result of this is that this nucleosome marches forward, and then the nucleosome that originally covered the Tata box region gets removed. Okay? So the Tata box region is now covered by this nucleosome, and then if you keep repeating this process, then at some point the promoter is going to be cleared of this nucleosome, and you have a Tata box region exposed. So I like to think of the mechanism of chromatin remodeling complex as this bulldozer. Right? So basically, this nucleosome is like a wheel. It's a wheel where DNA can turn around like this, and the chromatin remodeling complex is like a shovel. Right? So it sits on this wheel, and then it's going to plow through all the nucleosomes sitting ahead, which leads to nucleosome removal. And this similar model has been proposed, in fact, 4 years ago by Roger Kornberg lab. So what I'm trying to conclude is that nucleosomes can sometimes promote gene expression, right? On the contrary to what people generally believe. And the reason why we saw the drop in gene expression level when we destabilize the DNA underneath this intervening nucleosome is because if you destabilize the nucleosome, then you create this empty DNA region, which is about 200 base pair long. Okay? And you can think about the persistence length of DNA, which is about 150 base pair long, and they're about the same order of magnitude, which means that this DNA is going to be pretty stiff. Okay? So although the transcription factor recruits the chromatin remodeling complex, it's going to be located pretty far away, right, with respect to the persistence length of the DNA, so that it will have a hard time reaching this nucleosome which is covering the Tata box region. Okay? So that's why we think this template, which doesn't have a nucleosome, is not going to give rise to very efficient gene expression. Whereas if you have a nucleosome that is formed in this region, then you efficiently bring these two distant points close to each other. So nucleosome, although it's a barrier to be removed, it also functions as a wheel on which the chromatin remodeling complex can sit and perform its function. Okay? So to summarize the second part of my talk, I just introduced you how we measure quantitative gene expression. And I also explained to you that the distance between transcription factor binding site and nucleosome is very important, and that could be modulated by nucleosome formation. So we think that nucleosomes sometimes can play an active role in gene expression. So with that, I'd like to acknowledge my group members who are very essential for me to carry out my research. And especially, I gave you 2 talks today. The first part was done by graduate student Tung, and the second part by postdoc Rasish. I also like my funding sources, Georgia Tech and Burroughs Wellcome Fund. And thanks for your attention. Yes?
Ariel, thank you.
Sure.
Can you give us a sense from the first talk, what's the relative role of the electrostatic binding of the histone to the DNA versus the bending energy?
Yeah, very, very good question. So as you know, all nucleosomes, they have the same histone octamer, right? So it's the same, identical histone octamer. The only difference is the DNA sequence. Now, DNA sequence is negatively charged. All DNA molecules are negatively charged. And that charge comes from the phosphate backbone, not from individual bases. So in terms of electrostatics, it's going to be the same in every nucleosome, right? Not much difference. So that's why we think that this elastic energy actually plays a very important role in differentiating nucleosome affinity between different sequences. Yes?
Can you partly rescue the gene expression by shortening the distance and still have a
Yeah, that's a very good question. So to test this model, right, one way is to, we shorten the distance between the two. Yeah, so if this is now positioned over here, then we should be able to recover the same gene expression. And there is going to be some non-trivial distance dependence here, right, because this looping, it has this peak at some length, some distance between the two ends. Yeah, so that's an interesting experiment that could further give support to this model.
And the other thing I was wondering in your first talk, the thermodynamic parameters that are most relevant with regard to the binding affinity would be on rate divided by off rate, right? And
Yeah.
we haven't talked about the on rate, and I could imagine that the on rate might be actually quite different depending on the stiffness, and that a lot of what you talked about is sort of related then to that parameter.
Yeah, so
much a lifetime of the complex. I mean, of course, that matters too.
yes, I mean, that's a very good point. Yeah, so nucleosome formation, and people have measured this something called J factor, which takes into account both on rates and off rates. And so the affinity is going to depend not only on the equilibrium ratio between the two. That's what you're saying, right? It's going to depend more on the on rate? Or you think it depends on...
I would say it's both, right?
It's both, yes, okay.
Because you have the on rate will probably critically depend on the entropy loss you have by having...
Yes.
And that
yes.
will be encoded into flexibility, which is a penalty from an entropic point of view. But then the delta H doesn't factor that much in with regard to that. That will depend more than on the binding interaction. That actually might be also sequence dependent to some degree.
Yes.
How it matches with the protein.
Yes.
On a sort of
okay.
microscopic level.
So I'm not going to be able to answer your question, but let me give you some thoughts. So nucleosome formation, what we study is DNA looping, intrinsic looping in the absence of histone proteins. And of course, nucleosome formation is going to depend on both, right? The on rate and off rate, because both rates depend on the flexibility of the DNA. However, we think formation is mostly on rate driven because you have this protein and then the DNA has to bend a little to form additional context, right? You don't need to bend all the way to form a loop. You can just bend a little bit and form new context around the surface of the protein. So once that contact is formed, which has to be very strong, we think that the off rate could be negligible. But
so in your experiment, you actually measured the lifetime of the looping complex.
yes.
And how does that relate now to the on rate of the histone DNA interaction?
So that's something that I sort of explained. So affinity is not something we measured. Nucleosome affinity is measured by other people. We measure the on rate and off rate. Mm-hmm. And the on rate can be related to bending stiffness of the DNA molecule by this first passage time calculation. And from that, we extract the bending stiffness, right? From the extracted bending stiffness, we then calculate the nucleosome affinity by applying the Boltzmann factor. And then we compare the two numbers, and it seems like the number we get is larger than the number that was measured. Yeah. All right. Sure.